Hello and welcome to D-Block's efforts at membranes. As you can see here, we have a newspaper and polystyrene foam construction, which includes the channel proteins, a phospholipid bilayer made up of two phospholipids, with the phospholipid head and the fatty acid tails being hydrophobic, heads being hydrophilic. Over here, we've got a beautiful pink arrangement of channel proteins and carrier proteins and phospholipids and cholesterol. This is contributed by the F block, and we've got another one over here which is in polystyrene and 3D again, hydrophilic and hydrophobic, showing how polar molecules are able to travel through channel proteins, and we have cholesterol there to stabilise and regulate the fluidity of the membrane, and we have some carbohydrates acting as receptors as part of the proteins. Over here we have a pun on the phospholipid heads, so we can see we've got some glycolipids and we've also got phospholipids with the phosphate head that's hydrophilic and hydrophobic tails. Got extrinsic proteins that only span part of the plasma membrane, as well as intrinsic proteins which span the entirety of the membrane. Over here we've got some more interesting cotton wool balls as the carbohydrate, attached to, again, phospholipids, so those making glycolipids, and also attached to some proteins so that we've got glycoproteins. We've got tails, fatty acid tails, and hydrophilic heads. Over here we've got a fully annotated plasma membrane, including some cytoskeleton made of microtubules and a carrier protein and a channel protein. The sequins are the carbohydrates used to make glycolipids and a glycoprotein. Extrinsic proteins can act as enzymes, they can also act as antigens, and intrinsic proteins can act as receptors, or we can also have proteins acting as carrier proteins and channel proteins. Over here we've got a beautiful newspaper arrangement, again with some lovely beads to represent the carbohydrate attached to these phospholipids with the heads and the tails. Some of the tails may be saturated, which means that they have full hydrogens and no carbon-carbon double bonds. Some may be unsaturated and they will have carbon-carbon double bonds in them. Over here we've got a beautiful chocolate array with some laces, showing that laces are the hydrophobic tails and the cholesterol. You've got a channel protein, and you can see that channel protein here is carrying out diffusion because we have a higher concentration up here flowing down this way to a lower concentration down here. So this is facilitated diffusion. We can also have simple diffusion, which is through the phospholipid bilayer down a concentration gradient. That's for polar molecules, for example water, but only because water is so small. Normally polar molecules actually have to go through channel proteins. We've got some extrinsic proteins which could be acting as enzymes, and an extrinsic protein here which could be acting as an antigen. Moving along again, we've now got a very colourful model. We've got some red fatty acid tails, we've got some beige phospholipid heads, we've got channel proteins and carrier proteins. All the intrinsic proteins run the whole or span the whole of the phospholipid bilayer. So those are our intrinsic proteins, and extrinsic proteins only span part of the bilayer, as shown here by these straws. So again, we've got some cholesterol, we've got an intrinsic protein, we've got extrinsic proteins, phospholipids with a head and the tails, and cholesterol, which is made up of 4 rings. And cholesterol in a plasma membrane is there to regulate the fluidity. Over here we've got, again, phospholipids with the head and the tails, cholesterol, glycoprotein, channel protein, carrier protein. Carrier proteins are used in active transport and facilitated diffusion. Channel proteins are only in facilitated diffusion. In active transport, the shape change that you see in this molecule is due to the binding of ATP. The shape change can only occur in one direction, hence in active transport you can accumulate molecules on one side against the concentration gradient and they don't flow back out of the cell. Over here we've got a beautiful green and brown arrangement of the phospholipid bilayer, showing the green as the tails, heads in the brown. You've got some intrinsic proteins that span the whole of the bilayer, carbohydrate attached, and we've got some little bits of cholesterol as well in there. Over here we've got a sweetie phospholipid bilayer and proteins. So we've got a plasma membrane made up of phospholipids which can melt at high temperature, proteins which denature at high temperature, glycoproteins which act as receptors and antigens and can also bind to other cells as part of the basement membrane to form tissues. You've also got extrinsic proteins which act as enzymes, cholesterol to regulate the fluidity of the membrane. Over here we've got some more extrinsic proteins and intrinsic proteins. Again, channel proteins can behave to allow polar molecules through, tends to be smaller polar molecules or charged sodium ions. They also pass through channel proteins if it's going down a concentration gradient. If you want it to go against the concentration gradient, then you're going to need ATP and a carrier protein to move larger polar molecules through. For facilitated diffusion with a carrier protein, that's always using a carrier protein and no ATP with a concentration gradient, and that's typically things like glucose, so larger polar charged molecules. Okay, over here we have our balloon model. So the balloon model incorporates a balloon as a channel protein, and the phospholipids are represented by these green tails and these blue heads, and we've got some cholesterol over here as well. Back over here we have a beautiful little model, a 3D model hanging around, just showing the different components of the plasma membrane, which includes your red balls as your hydrophilic heads, tails, then you've got your channel protein, your carrier protein, so a glycoprotein and a glycolipid, and these little blobs are your cholesterol. And that's everything for the phospholipid bilayer and proteins, which together make a plasma membrane, and it's called a fluid mosaic membrane because it's fluid, so things can move around each other but only in the same plane. They don't move up and down like that because the heads don't like the tails, so they never come together. They just move around each other, so that's the fluid part, and the mosaic is the arrangement of these proteins within that phospholipid bilayer.